All right, what you've all been waiting for, the data analysis and graphing video. I know you're all amped up. In this video, we're going to talk about a lot of the skills that you are going to have to apply to the AP Biology exam content. And this is a very important part of the exam. I would say your ability to just look at data and determine trends, determine appropriate graphs, graphing, determining if something's statistically significant, all of that is going to be probably about 20 to 25% of the test. So this first basis that we're building is really important, and just be aware of that. I know because sometimes it'll feel a little bit dry. I'm going to try to make it as exciting as possible, but these skills are vitally important for the AP Biology exam. So we'll start off with this. This is a hypothetical question. Which fertilizer do you think works best here? So I'm going to give you a second to look at the data, look at the graph here, and I want you to determine which you think works best. Okay, most of you would probably say F2, right? This bar right here, correct? Now, I'm going to give you a little bit more information to see if maybe you come up with the same conclusion here. This is the raw data that I use to construct those bar graphs there. So I want you to look at these groups. The control group, in this case, received a sham fertilizer. This is the classic determining different fertilizers' effects on plant growth, right? We have fertilizer 1, 2, and 3. Control group is a fertilizer like a placebo fertilizer. They wouldn't receive any additional nutrients, things like that. So look at this data and see if you change what you think, okay? All right, so when I look at this data, there's a few things that I notice, right? There's a few things when we look here. The control group's pretty consistent. Average is right around 5.8, right? We have F3, which was the highest, or excuse me, F2, which was the highest. But what do you notice about F2 that's interesting and unique compared to like F3, for example? F2 has what's called more spread in the data. You have these wild outliers, 18.1, 12.4, and then you have some really low values like 2.5, and then you have a few that are right in the middle. Versus F3, which is very consistent, right? You can see there's not a lot of spread in that data, right around each one, right around 8.7 approximately, right? So if we look at this now, we determine, well, which one do we think is the best now? I would say F3 is the best because of that spread. Maybe this is just a random data point. Maybe that plant has a genetic difference that caused it to grow more. Who knows? But F3 is a more reliable indicator, right, of the effectiveness of the fertilizer because it's very consistent, okay? So, but the problem is we cannot just look at data and determine these things with our eyes. We have to do what are called statistics. We have to analyze the data using computer programs because we want to, as we talked about in the previous video, you have to worry about bias. Bias can creep in even when we don't want it to, right? We don't care about opinions. Opinions can change from person to person. We need a concrete number to determine whether or not there's truly an effect of an independent variable on a dependent variable. So data, in terms of what we look at, what we collect, right, with our senses, is nothing without statistical analysis in biology. We can look at it and see trends, but we cannot rely on our biased opinion most of the time. So remember, statistical bias is anything that leads to a systematic difference between two parameters of a population. This is more of the textbook definition, right? It refers to the flaw in an experiment design or collection process which generates results that don't accurately represent what we're actually testing. That can be purposeful or not purposeful, right? Our goal in science is to remove that bias. So the first thing we're going to talk about are simple descriptive statistics: mean, median, and mode. In mean, that is simply just the average. And what you're doing is you're collecting data on some sort of variable where you collect that from a lot of different individuals. And then we are taking the average of them, taking the total, dividing by the number of points we have, and that gives us our average. Our median is a little bit different. That's our middle value in the data set. And means and medians give us different information. So if we lay out all of the numbers on a number line that we collected, or all of the data points on a number line, the median would be the one that is smack dab in the middle, and then the mode is the most common value. An interesting difference between mean and median would be like mean average household income in the United States. The mean and the median tell us very different things, because we have a lot of people in this country who don't make a lot of money, and we have only a few people that make tons of money. That actually skews the average up higher. A median is probably a more accurate value of the true middle class of the United States, right? Not the mean or the average. Mode is the most common value, so that's if you're seeing it consistently over and over and over again. That becomes more relevant for things like histograms and like data that is like part of a bell curve, that kind of thing. And they all give us different information. Quartiles relate to median too. So if we take all of our data sets and there are 3 different numbers that divide a data set into 4 parts. 1, 2, 3, so 3 different divisions create 4 parts. The first quartile is the lower quartile. The lower quartile is going to be the lower quarter of the data, would be within that lower quartile. The second quartile would be the up to the median, right? So the median would be the second quartile. The third quartile is the upper quartile that divides between the upper third and the top fourth of the data set. And here's showing you kind of what I mean by that. If we laid them all out, this is where the quartiles, the 3 that divides into 4 parts, and here shows you actual data that would tell us what is the first quartile, the median, and the third quartile. So we can actually visually represent these descriptive statistics really 2 different ways that you could see in the AP Bio exam. They can always throw in other ones, right? But these are 2 ones that they tend to want to focus on, and I've seen them many times on the AP exam. So those are box and whisker plots, and also bell curves, normal distributions, or histograms. So first, we're going to talk about a box and whisker plot. A box and whisker plot will visually represent a full data set in a way that we can kind of just look at it real quick and determine what is the spread there, how close are the points to each other. And the way you construct something like that is you have the median, which is the line in the middle of the box, right? And then you have the third quartile, which is up here, the top of the box, the first quartile, which is the bottom, and then you have the lowest point and the highest point as your whiskers. Sometimes they also have outliers that they would throw out that they include on there as well, but usually they don't. And that's how you would construct a box and whisker plot if the AP Biology exam asked you to do that. Here's showing you an example of a bunch of box and whisker plots, all looking at, this is actually related to football, at 40 speeds of different positions on the football field. And as I say at the top, we cannot determine if there's significant differences. We'll talk about what that means when we get into error bars soon. But we cannot determine if there's actually differences between 2 things here. This is more just to visualize data, at least in terms of statistically significant differences, which is important in science to do. Again, I'll talk about that later. Here's an AP Biology exam from an FRQ from 2023. It shows you a CQ value, which don't worry about what that means, but you can see here the different developmental stages and different genes that are turned on and how widely expressed they are in the different cell types. Like one cell type has different effects. Like we can't say they're statistically different, but we can say where the mean, the top quartile, the lower quartile, and then the distribution of the data there. Scene change. Okay, so another way we can look at that type of data of means, medians, modes, that kind of thing, is a normal distribution or a frequency diagram or a histogram, which we're going to look at in a second. Many different population data sets, if we're looking at traits, form what's called the normal distribution or a bell curve, where most individuals that have like an average kind of version of that trait, but then you have people that are more outliers. The more rare those traits are, those are going to be the extremes of that trait. Think of it, things like height, right? Most people, right around for males, like around 5'10". That's most people in the population. How many people do you see that are 6'5", 6'6"? Much more rare, right? How many people do you see that are 5 foot that are males? Much more rare, right? They're extremes on either side. So that would be an example of a normal distribution. Most of the data lies really close to the mean. As you can see here, this is showing you about, this is called the 1 standard deviation from the mean. I don't know if you've learned standard deviation before, we're going to talk about it in this video a little bit. Then there's 2 standard deviations from the mean. 95% of points fall within that range. And then there's the extremes, which are the last 4%, and the bottom 2% and the top 2%. Those are called tails on either end of the normal distribution. And you can see this is looking at height distributions of males and females. That you can see how normally distributed that is as well. Now, a histogram is basically a bar graph version of that bell curve. It represents usually frequency distributions. That means like frequency is a percent or the decimal version of a percent. We're going to be dealing with frequencies a lot in AP Bio. And you're going to have frequency on your y-axis and then the trait in question on the x-axis. So this is looking at like grade distribution in a population. You can see here how many people got between 0 and 9, 10 and 19, all the way up to 90 and 100, where most people fall in this average range. Now you're like, that's a really low grade distribution. Don't worry about that. That's not how I distribute my grades. But you can see that's a very normal distribution. Most people are right around the average. You can also skew distributions as well. You can be skewed right or left, as you can see here. This is showing a skewed right distribution. You can also have bimodal distributions where you have a peak, a drop, and a peak. Those are going to be very relevant when we get into evolution and looking at the evolution of specific traits in a population and the frequency of those traits. Another thing that you have to learn is variance. And variance is basically a measurement of how far each point differs from the mean. If you look at the statistics, now you're not going to have to calculate variance. At least I don't think so. It does appear on the AP Bio exam on your reference table sheet. But you can see this is how each point, xi, how far it is from the mean, we square it, and then we divide it by the total sample size minus 1. So the further each point is away from the mean, the larger the variance is going to be. If everything is right around the mean, your variance is going to be really low. We do not want to have a lot of spread in our data because it's a little bit less reliable. Why are there data points all over the place, right? Everything should kind of be consistent. That's what we'd prefer, but obviously real data doesn't work that way. So we have to measure the variance. Standard deviation takes your data set and it'll standardize it. So it compares it to like all the other data sets in terms of those things that would be measured. If there are a lot, again, standard deviation is just another way we can measure spread. It takes your variance and takes the square root of it, essentially. That's all it does. If there are many values that are far away from the mean, there's more spread, there's higher standard deviation, and there's also more variance. If they're all close to the mean, you're going to have low standard deviation, low variance. So there's not a lot of variation in the data. The way standard deviation works, 95% of all data points will fall within 2 standard deviations of the mean, and 68% of all points should fall within 1. So it kind of takes our normal distribution and determines for our data set what that normal distribution would look like. Now, this is pivotally important. We talk about this in AP Bio all the time. Standard error is something that I'm going to bring up in every single unit because this tends to be the way we measure significance in AP Biology. Now, there are some problems with that, but on the AP Bio exam, we're looking at standard error and error bars to determine significance, if we can determine if there's a significant difference between 2 data sets. So it takes the standard deviation and divides it by the square root of the number of samples taken. The larger your sample size, the smaller your error will be. So if you have a standard deviation, if you have a very large sample size, it will actually decrease that standard deviation as you increase the sample size. And that makes sense because if we're collecting a lot of data that's all over the place versus a little that's all over the place, we're not really sure whether or not that little data or that little sample size is not just statistical noise. But if we're seeing it consistently over and over and over again, it's just there's a lot of variance in that data, we can kind of say, okay, there's a little bit less error. We're a little more sure that this is an actual representation of what the mean or what our mean is or what our data set is. Okay. When we graph our mean, you can do with bar graphs. It can be on points, right, for line graphs, and we'll talk about that. You can add error bars. And error bars are plus or minus. If you do plus or minus 1 standard error of the mean, there's a 68% chance the population mean falls within that range. If there's 2 standard errors of the mean, which is what we do for error bars always, just so you know, on the AP Bio exam, at least standardly, they'll be graphing plus or minus 2 standard errors of the mean. There's a 95% chance the true population falls within that range. So what you're basically doing when you create error bars is look at, you have your mean, and then you put a plus or minus value to where we think the true mean falls, and we're 95% confident it falls within that range. If there's no error bar overlap, we're pretty confident that those are two different samples, that their means are different. But if there is overlap, we cannot say that that is the case, and that's how we determine significance. So if you can see here, now this is just one-time standard error of the mean. Technically, standard error should be these error bars should be double that. As I said, you can do one, but AP Bio wants you to do two. So this is showing you how you would graph those error bars. If our mean is 10.5 and our standard error is 0.25, then there is a 68% chance that the true mean falls between 10.25. I take 10.5 and subtract 0.25, the error, and then I add the error on top of it. That's 10.75, and that's where I put those points. Or there's a 95% chance that the true population falls between 10 and 11 because I took that error, what was 0.25, I multiplied it by 2 because that's 2 times the standard error. And then, so I would add 0.5 and subtract 0.5, that's our range. That we're 95% sure that our mean falls in between 10 and 11. Okay, so this is how error bars would look when they're graphed versus control and experimental treatments. Now, we're going to pretend that those are 2 times standard error. I know they're not in this case, but if there is overlap between them, as in experiment 2, you can see that this error bar overlaps this error bar. I cannot say with a 95% confidence that those are actually different means. Because what if the treated value was all the way to the top and the control was all the way at the bottom? Now their relationship has shifted and flipped, right? Whereas in experiment 1, again, pretend it's 2 times standard error of the mean. Experiment 1, you do not have overlap there. And we can confidently say even if that control group was all the way at the bottom of that range and the treated group was all the way at the top of that range, there's still a difference, therefore they're statistically different. So as I mentioned previously, error bars are not only found on bar graphs, we can also put them on line graphs as well. On line graphs though, and I'm going to go over graphing at the end of this video and the different types of graphs when they're appropriate, but a line graph versus a bar graph is usually determined by the type of data you're collecting. If the data is continuous, time, like pH scale, amount of substrate, concentrations of solutions, that means like if I can get information between 2 points, like if I have like a measurement of rate at a pH of 2 and a measurement of rate at a pH of 5, if I want to know the pH of 3, I could actually go in between and get information on that rate because the rate would be between those 2 points, right? So that's when you would use line graphs. But as I said, you can still put error bars on line graphs as well. And you can see there's clearly a statistical difference. Initially, there's no statistical difference between these two. And finally, eventually, after they're isolated, you can see that there's a significant difference between these isolated the spots on the guppies, the male guppies in this population versus this one. And it probably has to do with isolating them and maybe, putting a predator in there versus no predator. So bar graphs use what's called categorical data. Again, I'll go over this again, but it's data that is not continuous. So like a group that receives, I want to look at the short-term or long-term effects of caffeine on use on sleep habits, right? So yeah, that's a time, but it's short-term versus long-term. Those are groups. I can't get intermediate information from there necessarily, right? At least not accurately. If we're looking at the difference between a group that receives fertilizer and a group that does not, there's no in-between there. So we can't get an intermediate value. So that would be a bar graph. And as I said, they tend to be categorical. So as I mentioned before, our goal in science is to determine if something is statistically significant, because that, if we find a statistically significant differences, those will be accepted by peers. Our threshold is 95%, or a 0.05 chance, or a 5% chance, not 0.05% chance, but a 5% chance that randomness could explain our results. That's our threshold. So 95% chance that randomness did not, we say we can be pretty confident that our data is significant. Now, where did that value come from? It is completely arbitrary. We've just determined that. Obviously, the lower that value is, the better. If there's a 1% chance that randomness can explain it, that's even better. If there's a 1 in 1,000, a 0.00 1 chance that randomness can explain it, it's even better. So our goal is to try to get that p-value as low as we can to be different. Now, again, we're not trying to do it. We want to just collect the data to determine if there's a difference. We do not necessarily want to prove or support. We don't say prove in science. We don't disprove things either. We just support with evidence. Don't ever use prove, by the way. Don't ever use it. Ever. Don't say prove. We don't prove anything. If you do, I'm going to take points off on your test or your quiz or whatever you hand in to me. Don't say prove. I will cross it out aggressively in red. We'll talk about the p-value. This is called the p-value, this percent chance. We're going to talk about that a little bit more later.
All right, so for hypothesis testing, what we're actually doing
is we are determining whether or not, using statistics, not just error bars, which is a visual representation, we're going to use statistics to determine whether or not there's a statistical difference between two things. Now, what we are going for is we're trying to get a value called the p-value, and the statistical program will spit out a p-value, and we're always looking for those p-values of less than 0.05 to determine if there's a significant difference between what we're testing and our control group. There's a bunch of different types of stats that we will learn. T-tests, ANOVAs, chi-squared tests, which is going to be the most important one in AP Bio. I am going to briefly go over it. Our first lab that we're going to do is going to be a chi-squared lab where we look at M&Ms, and it will, I think it's the hardest to understand, but we're going to literally use it all year. So by the end of the year, you should have a pretty comfortable understanding of how to use in the chi-squared when it's applicable. And we'll also talk about something called correlations and linear regressions. A correlation isn't an actual hypothesis testing statistic, but a linear regression is. We're just going to briefly discuss those and what they mean. So, vocabulary you need to know for hypothesis testing. You need to know what a p-value is. That's your probability value. This describes the percent chance that randomness could explain your results. Remember, the threshold we are going to use is a p of 0.05. If we are greater than 0.05, there is not a significant difference. If the p is greater than 0.05, or if the p-value is less than 0.05, you will say there is a significant difference. This will also relate to the null hypothesis. If your p-value is greater than 0.05, because that's not significant, you fail to reject the null hypothesis. And remember, what's a null hypothesis again? No effect, right? There's no effect of the independent variable on the dependent variable. When your p is less than 0.05, we can say that there's a difference. There's a significant difference. We can say that there is an effect. Notice I didn't say prove anywhere. Don't say prove, okay? The degrees of freedom value is something, it depends on the statistic. This value that deals with the number of comparisons you make or the number of data points that you have in your data set. It's usually the number of data points you collect minus 1 or 2, depending on the type of test you're doing. For chi-squared, which is going to be the only one you're going to have to know it for, it's always the number of comparisons you're making minus 1, but we'll get into that when we get to that step. So remember, p-values of less than 0.05 means that it's significant. There is less than a 5% chance that randomness would explain the data we collected, and we consider that statistically different. That is our threshold value. P of less than 0.05, not greater, less, or equal to. If it's equal to, we also consider it significant. Okay, so you want to know that. I don't know where I got this from. I typed in something about like p-value cartoon, and this is what came up. This is what's on the internet. I don't know. First test we're going to talk about, again, I'm not going to really make you, I'm going to have you guys do an example of this. You do not have to actually do t-tests on the AP Bio exam. It could give you t-test data results if they do it, but I'm just trying to get you to understand how different types of data and how we can test those different types of data through different circumstances and using different tests, right? Because there's different types of data and we need to test them differently depending on those types that we have. So a t-test, like this looks super complicated. You don't need to know how to calculate it. You would type this into a spreadsheet and it would do it for you. And this is like the classic hypothesis testing statistical analysis. If I want to test the effect of fertilizer on plant growth, One set will get fertilizer. We'll measure the average height of all the plants that get fertilizer after a certain amount of time. The other does not. That's the control group. We're going to average the average height of that, and we are going to put that into a t-test, and it'll spit out, is there a statistical difference between the two? So here's an example of a t-test data set, and also the results from that. So we have subject ID. These are students, and we're testing the effect of teaching on achievement. So you can see each of those are individual students. They don't have actual names associated with them, but they are linked to their pretest and post-test grade after, I would assume, several weeks of instruction. So there's their pretest grade, number 1, 90.563, post-test grade, 110.642. And you put that and you go down the whole entire thing. Now, you would use a bar graph to graph this. You would take the mean of both, because that's what we care about, it's the mean of both, and then we would graph those means, and then we would add error bars to them. This would also, if you notice, a p-value of 0.002. Is that less than 0.05 or greater than 0.05? It's less than 0.05. That means there's a statistical difference between the pretest grades and the post-test grades on average. That means that learning happened, right? They did better on the post-test than they did the pretest. And if you look at their means, right, the pretest mean was 97.06. The post-test mean was 107.83. We have a standard deviation calculation. We have the standard error of the mean. And even if we add 2 times the standard error of the mean and subtract 2 times the standard error of the mean, you're still going to get close, but there's going to be a statistically different value there. Okay. So we would reject the null hypothesis that there is no effect on teaching on achievement. And in this case, we support our alternative hypothesis, which is that there is an effect of teaching on educating on achievement on a pretest versus a post-test. There's something called an ANOVA, which it's like a t-test, but it compares multiple different data sets. It's a little bit more robust. It's more complicated. If it ever says ANOVA, sometimes they'll just write it was analyzed using an ANOVA on the AP Bio exam. That's just what that means. It's not anything you have to know, as long as you understand the p-value point, that there's a difference between those two data sets based on the p-value of less than 0.05. Another statistic we look at, now this is not a hypothesis testing statistic, it's called a correlation. They sometimes make you graph correlations in the AP Bio exam. This is to determine if there's simply a relationship between 2 variables, like BMI and blood pressure. Do people that have higher BMI have higher blood pressure? No, some people that have higher BMI may have a lower blood pressure. We're going to put it all in a plot. We're going to put one variable on the X, one on the Y. We're going to put the one that we think BMI affects blood pressure, not vice versa. So we'll put the BMI on the X-axis. We'll put the blood pressure on the Y-axis. And we'll just point each individual person and their stats of both BMI and blood pressure. And what you're trying to do is you determine, is it a perfect correlation? Is it a weak correlation? Is it positive or negative? Is there, as one goes up, the other goes down? That's a negative relationship. As if one goes up, the other goes up. That's a positive relationship. And these values of the statistic range from -1 to 1, with 0 being no correlation, 1 being a perfect positive, and -1 being a perfect negative correlation. You would graph this by putting data points on a scatter plot. As I mentioned before, I'm going to show you what that kind of looks like in a minute. For example, we wanted to test the effect of height on IQ. We would collect data on the individual's IQ and that individual's height. So those are paired together, and we'll graph them as a point on height versus IQ. And we'd repeat that with 400 other people, and we get a nice sized data set. We'll put it on a scatter plot, and we can kind of see if there's a relationship there. So here's showing you an example of one that appeared on the AP Bio exam just a couple of years ago. They wanted you to graph a graph showing a correlation between the number of strand breaks and the average number of crossovers. So when you did this, you graphed your points. They labeled each individual point here. They had the average number of crossovers that was impacted by the number of strand breaks. Again, you don't know what that data means at this point or even what those processes are. Don't worry about that. We want to see if strand breaks and number of crossovers were related. You can see here, this is a pretty good, pretty linear relationship there. That would be a very high positive correlation because they almost all fall within a best fit line between those points. But that's an example of one that was on the AP exam just a couple of years ago. Here's showing you again more points, but what it would look like having strong positives, weak positives, strong negatives, weak negatives, and then no correlation whatsoever, as you can see here. So here's a strong positive, strong negative, all those points. You can almost like imagine them. You can see a really nice kind of either going up or going down. The weak positive, you can see it goes up, but like there's definitely outliers here. There's a moderate negative, a little bit more spread out than the strong negative, but that would still be like a negative, maybe like 0.7 or point whatever. Yeah, probably about a 0.7. And then this would probably be like a negative 0.4. And then this would be almost 0 because there's no relationship there whatsoever. You cannot hypothesis test here. You just determine if there's a relationship between two variables. And now we get to our chi-squared test. This is the test that is used to see, and we're going to talk about this the most because we're going to see it over and over and over again on the AP Bio exam and throughout the year. This is used to see if counts data, so tallies data, the number of individuals of one thing versus another, differ from what's expected using probability. So a great way to think about this is with a coin. If we flip it 100 times, we would expect it to be 50 heads and 50 tails. And if we didn't get that result, if we got like 70 and 30, we'd think something was kind of up with the coin. There's a bias towards the one head. It's not a perfect 50-50 split. So for what we would test in this case is this coin rigged? And we'd compare it to what we'd expect to get based on probability versus what we actually get. The further we deviate from what we expect, the higher probability that there is actually something going on there, that the coin's rigged, right? We can apply this to a lot of things. We're going to apply it to genetics, we're going to apply it to evolution, or we're going to apply it to mitosis, behavior, all different places. So this is the math. This is coming off the actual AP Biology reference table. Chi-squared is equal to the sum of all observed minus expected squared over expected. So we're going to try to solve this using the coin toss example that we just did, and we got 47 heads and 53 tails. Now I want you to look at that data. It's supposed to be 50-50. We got 47 and 53. That is absolutely probably within the realm of random chance, and it doesn't mean that the coin's rigged or anything. It could have just happened that way, right? All right, let's look at some actual data here, okay, of that exact scenario we just gave you. So if you can see here, we have our observed and our expected. We put them into the chart. 47 heads were observed, 53 tails were observed. We expected to get 50-50, right? So we take observed minus expected of each one, -3, +3. We then square that, as it shows here, of each one, 9 and 9. We then take that and divide each one by the expected, which was 50, and we get 0.18 and 0.18, and then that sum of all, that E, right? That is added, you add the two together, you get 0.36. That's called our chi-squared value, or our calculated chi-squared value, okay? And we have 1 degree of freedom here because there's two possibilities that we have, either heads or tails. So we subtract 1 from the number of possibilities, our degrees of freedom is 1. So notice there we get a p-value of 0.36. I'm going to do something real quick. I'm going to modify this just slightly. So if you look, I'm going to escape out of this. I'm going to change this now. What if now we change this to 35 and this to 65, right? Now, when you look at that data, it looks like it's definitely biased towards tails. Like there's something with the tails that's coming up more often. We'll still expect 50-50, but notice what happens to those observed minus expected values. Now it's minus 15 and 15 in the other direction. We divide, or now we have to square 15. 15 squared is, let me just calculate it real quick, 15 squared is equal to 225. 225. Now we divide that by 50. We get 4.5 for both. 4.5. 4.5. And now we have a value, a chi-squared value of 9. So remember, we're going to look at this and compare it to what we got before, that 0.36 value. And what does it mean when we look at the chi-squared chart? So here's a chi-squared chart. This is the one we're going to use in the AP Bio exam. It's a little bit more simplified. This is an actual chi-squared chart. I'm going to kind of show you both how they actually work here. So if you look here, remember we said we had 1 degree of freedom because it could be heads or tails. -1, that's one possibility, or 1 degree of freedom there. And our threshold value is always a p of 0.05. Notice it says p-value here, 0.05. 1 degree of freedom, 0.05. If we go to the chart, that value that is on this chart is called the critical chi-squared value. You're seeing whether your value is less than or greater than or equal to it, or if it's less than it. So if the same thing's true here, you'll see the values are the same. 1 degree of freedom, P of 0.05, 3.84, 3.84. Okay? So when we look at that value, we then have to determine, is our value greater or less than that? The first example, where it didn't look like there was very much variation, we got a value of 0.36. 0.36 falls on this side, which means that there is, that your p-value is greater than, as you can see here, 0.05. And that tells us that there is not a statistical difference, or we will fail to reject the null hypothesis. In this case, the null hypothesis, which remember is no effect, is that there is no difference from what we'd expect to get. Okay, if your value is above that threshold of 3.84 in this case, which the second example was, right, if it swung 15 and 15 in either direction, we got a value of 8. Notice that falls in this range. There's less than a 0.025% chance that randomness could explain it. That would tell us that there is an actual difference there. Okay, so that's how you would do this, and you can see that here too, right? We're falling way below this value here, therefore there is a statistical difference. We would fail to reject the null hypothesis in that second scenario. All right, so a common misconception that you tend to see, right, with chi-squared is not understanding the difference between a p-value, degrees of freedom, the chi-squared value you calculated, and the critical chi-squared value. Okay, so I'm going to talk about each thing. So when you do the chi-squared statistic, right, you go through, you do all the math that I went through before, right here, right, you go through this and you got 9 in this case. That value is your calculated chi-squared value, all right? We want to see if that calculated chi-squared value is greater than or less than the value on the chart at a specific number of degrees of freedom and a p-value of 0.05. So if we look at a p-value of 0.05, which is our threshold value, don't get that confused with the critical chi-squared value, at 1 degree of freedom, our critical chi-squared value is 3.84. So the critical value is the one we're comparing ours to. If we are greater than that value, we reject the null hypothesis. If we're less than that value, we fail to reject the null hypothesis. Values greater than the critical value correspond with low p-values. So large chi-squared values correspond to lower p-values. Lower chi-squared values correspond to higher p-values, okay? And remember, we want lower p-values are better if we're trying to determine if there's a statistical difference from what we'd actually expect. All right. Now, this is going to be really important to talk about, which is like, what are all the different graph types you could be able or be asked to construct on the exam? There's always one graph construction question. It's the second FRQ on the extended response section. And you could be asked to kind of do any of these graphs. The most common ones are going to be the bar and line, bar being the most common. I'll go over the parameters of those. There was an XY scatter, as I mentioned a couple of years ago, and the box and whisker plot that I went over before. Again, I'll show you examples of each one. So for a bar graph, that's categorical data. We mentioned that. That is like this group gets a treatment, this group gets a treatment. We average all of the individuals that received this treatment and all of the individuals that received this treatment. Whatever we're measuring is our dependent variable. And usually it'll involve error bars, and you just put a bar up to the mean, right? That's a classic bar graph. An example of this, number of individuals that like Pepsi versus Coke. I don't get any information about a hybrid between Coke or Pepsi, right? What's the average number of individuals in each school that likes Pepsi or Coke? I don't know. That'll be a bar graph because you don't get any information from the in-between. Or the short-term or long-term effects of caffeine on the number of visits of bees. Short-term and long-term, those are your two groups, right? That's categorical. Or if it says individual 1, 2, 3, or group 1, 2, and 3, and you cannot get information from the in-between in those groups, right? You cannot do that. That would be a bar graph. And you will always have to graph plus or minus 2 SEMs on these on the exam, the error bars. Line graphs, that's continuous data where you can get information from the in-between. Many times it's either time, temperature, concentration, or pH on the x-axis, or concentration of molarity or something on the x-axis. You probably will have to graph error bars on those points as well. You just go plus or minus over the top and bottom of the point, but you would connect the line because you can get information from the in-between on those points. If they ask you, if it feels like it should be continuous, but they still ask you to do a bar graph, you need to, on the AP exam, make a bar graph. Just don't ask questions, do what they tell you to do, okay? Then there's the XY scatter. The keyword there is correlation. You're looking for the word correlation in the prompt. If you see that, you will be doing the XY scatter. Do not connect the dots in an XY scatter because they're just random points. They're not related to each other. This person with a BMI of this and a blood pressure of this is not related to this person with a BMI of this and a blood pressure of this. So I wouldn't connect the line between them. I don't get information from that. Remember, those are looking for correlations. They're looking for relationships between two variables. And then there's the box and whisker plot. Remember, that looks at the overall parametric data. Well, it could be parametric data, but you're kind of looking at descriptive statistics there. That's where you graph the first quartile, the third quartile, and the median, which the median is the second quartile. That's the line. They'll put that in the middle of the box. And then the outliers are your whiskers. Here's showing you examples of each from the actual AP exam. This is not a box and whisker from the AP exam because they really only have one other one that I've seen recently, and I already put that on here. So this is just another example of some biological related box and whiskers. So here you have your medians, the line, you have the third quartile, you have your first quartile, and then the top point and the bottom point. You can see here, again, mean, first quartile, down here, third quartile, and then you have your outliers. Mean, third quartile, first quartile, and then you have your spread there. This is a bar graph. Remember the independent variable here, the effect of exercise on blood prolactin levels. You can see there is absolutely no statistical difference between any of this stuff here because all of the error bars just overlap with each other. Remember, to determine statistical difference, we're determining are the error bars overlapping or are they not. If they're not, we can say they're different. If they are not considered different. So there's no effect here. So like you would want to say, oh, there's after 1 hour, there's more prolactin with exercise. Nope, no, there's not, because these error bars overlap. We can't say that. Or without exercise, there's less prolactin. No, there's not. We can't say that. The error bars overlap. In fact, the error bars of all of these overlap. There's no difference between any of them. And here's a line graph, again, showing you error bars. There's 3 different lines here, mRNA 1, 2, and 3. Again, don't worry about what any of that means. These are concentrations of different mRNAs over time at different temperatures, as you can see here. See, this one goes up and then goes down. This one goes up and then goes down a little bit after it. This one kind of stays low the whole time, goes up a little bit here. But we can figure out, well, at 4.5 hours, how much mRNA was there? Well, probably somewhere between these two, right? So that's why we use a line graph in that case. And here's showing you an example of an XY scatter, beak depth of birds compared to beak length of individual birds. So each of these points was an individual bird and looked at their beak depth and length, and they also had different species here, looking at how those different species compared with their beak depth versus length. Other graphs you'll probably see, right? Pie charts. You'll see those. We'll see them in mitosis actually a lot with the cell cycle. That's just based on percentages, like what percent was this, what percent was that. You do what percent of students got a 1, 2, 3, 4, or 5 on the AP Bio exam. You do that as a pie chart. All around is 100%, and then each piece of the pie is a smaller percent within that. A histogram, again, that it's a normal distribution, or it doesn't have to be, but it relates to frequency. You graph frequency compared to some sort of trait. This is showing you like beaks of finches and beak sizes in different species. You can see this is a bimodal distribution here. This is a pretty normal distribution. This would be a kind of left skewed distribution, or excuse me, right skewed distribution here. You can see how it's not perfectly normal. It's kind of skewed more towards the one side than the other. But if you see it, that's what that means. I don't know if I've never seen them make you draw a histogram, but they could technically, and they would give you the data for that. We'll be looking at different examples of that as we go. So that is everything on biostatistics. you are now going to do an activity on Google Sheets, where you learn how to do some of these stats. You do not have to do the stats on the AP exam, with the exception of chi-squared, but it also teaches you a little bit of spreadsheeting. It teaches you how to use them. That's important for college. So be aware you're not going to actually have to do that type of analysis, but you will have to look at data sets and figure out, well, what is the most appropriate type of statistical analysis to do in each case. You're going to have some where I guide you through how to do it, and then there are going to be a couple of unknowns that you're going to have to kind of do on your own. If you are stuck at all on any of these, feel free to email me over the summer. I will be doing a review session on Google Meet at some point in August, towards the end of August, closer to school. So if you have any questions on any of the summer assignment stuff, we can go over it. So I hope this was helpful. I hope you learned some stuff, and good luck on the summer assignment.